Okay, let's go ahead and start. So we're starting a new section today, so just to kind of give you an idea of where we've been and where we're going. We started off the semester talking about how prokaryotic cells were put together. Then we moved into how they can move and how specifically they move towards nutrients, so the section on chemotaxis. Then we moved on to, once they got to that point by chemotaxis, how do they take up the nutrients? So we talked about active transport. And then once they had taken up those nutrients, how do they break them down? How do they produce energy? How do they make some of their molecules, like amino acids and such? And so how do they use those nutrients to create energy and precursors? And now what we're going to do is we're going to talk about how it is that they use that energy, although we're not going to talk about the actual energetics, but how is it that they use this to grow and to divide? And so we could do some mathematical manipulations and calculate generation time for bacteria. We're not really going to do that, but the time to double, or the generation time for bacterial species, varies tremendously. There are bacterial species that under optimal conditions in the laboratory can double their population in 13 minutes. There are others that take days or weeks. Mycobacterium tuberculosis, the bacterium that causes, obviously, tuberculosis, is one of those really slow-growing bacteria. It was one of the reasons why people couldn't identify tuberculosis as an infectious disease for many, many years, because they couldn't stain the bacterium and they couldn't grow it because it took so long for it to grow. So there's a tremendous range, but by and large, most bacterial species replicate really fast. So bacterial growth curves are typically shown on a semi-log scale. So if you look here, you have two different ways that you could plot bacterial growth over time on the x-axis. You could plot it on an arithmetic scale, where this binary division goes from 1 cell to 2, 4, 8, 16, 32. And you recognize that as those numbers get larger and you double them each time, you're eventually going to reach this asymptote. So what's generally done in terms of graphing the number of cells is to, rather than put them on an arithmetic scale, is to put them on a log scale. So that would be shown here in the red line. So that as those populations double and then double again and double again, you can see it as a straight line growth. So typical generation for E. coli, for instance, is 20 minutes. But other bacteria can take days or weeks. But this is in the lab. So in the lab is certainly not the real world. So it's kind of unfortunate that we do most of our experiments on bacteria in the laboratory, because it's oftentimes not reflective. E. coli points this out very well. In the laboratory, the experiment that you guys did this week, if we did the calculations, we would see that E. coli replicates, doubles its population roughly every 20 minutes. But in its natural environment, where it prefers to live in the colon, it takes 12 hours to replicate. So it's obviously a very different environment that the microbe lives in its natural setting. So laboratory is a little bit unusual. So we could go through all of this, and there is some value to going through all of this. I don't personally see the value in going through all of this. Those of you who are math majors, double majors, CAMS majors, my apologies. This is as much mathematics as you're going to get. The value to this, though, would be very obvious if you had a question like this, where a Taco Bell manager has accidentally inoculated 5 E. coli cells into the burrito meat. E. coli replicates every 20 minutes, so after 4 hours, how many bacteria would there be in that burrito meat? So here's an equation that you can calculate it, and stunningly, it's 10 trillion cells in just 4 hours. So it kind of shows you the power of, I shouldn't pick on Taco Bell, I just happened to find this online and it cracked me up this morning, so here it is. But in reality, this is kind of what you were doing in the laboratory this week. Or not kind of, it was what you were doing in the laboratory. It was generating a growth curve. And again, growth curves like this are entirely laboratory artifacts. And it's kind of an unfortunate thing. So let's go through and talk about the different phases of growth in the laboratory. So we'll start off with what's known here on the left, where this, shortly after this culture was inoculated, what's known as the lag phase 1. So during this lag phase, you can see over time there's very little increase in cell number. You can't tell that there is actually a cell number increase because this is on a log scale. So small differences are not very obvious, so it looks like a flat line. There is some increase in cell number, but it's pretty slow. Basically, what's happening during this lag phase is the bacteria have found themselves in a new environment and they're just going, holy cow, what do I do now? So basically, this is just a period of time where the bacteria are preparing to replicate. So they may have to synthesize new enzymes. They may have to get rid of toxic materials depending on the culture that they come out of. So this lag phase can be quite long or it can be quite short depending on the condition of the bacteria used to inoculate this culture. But after some degree of lag phase, you enter into what's known as exponential growth or logarithmic growth, or this log phase, shown as number 2. This is, on a logarithmic scale, a straight line. This is when the bacteria are fat, dumb, and happy. This is when they are at their enzymatic best. They are at peak performance. They are replicating as best the temperature and the conditions will allow them to. And so they continue on this logarithmic phase. Now, this is where most microbiologists sample bacteria in the laboratory to do their research on. When you look through the literature, you'll always see cells were grown to mid-log phase. So what they do is they grow the cells to around here, and then they do all of their enzymatic studies, their genetic studies. But in reality, this is a very unusual situation for a bacterium to find itself in the natural world. This would have to be a situation where one or a few bacteria found itself in a new environment with no competition, with perfect conditions, and just started going wild with no competition. So unfortunately, a very artificial situation. The next phase that the cells would reach is the stage that I think is actually more reflective of the conditions that virtually every bacterium existing in the natural world, whether it's in our intestinal tract or in Lake Metoka or the Kremdel, probably faces. This is the stationary phase. So here, in stationary phase, by definition, you have an equal number of cells divided as are dying. So there is no net population growth at this point. There are cells that are dividing, but they're just equaled by the number of cells that are dying. And this stationary phase makes sense because it always follows this very intense logarithmic phase of growth. So what's happened during this phase is the bacteria have now begun to use up the resources, and also, very importantly, they have secreted a lot of waste products. So no one, bacteria or us, like to live among our waste products, and so these things become toxic, and nutrients become rate limiting. So the log phase can't last forever, and it gives rise to the stationary phase. So this is why, like in the previous slide, I said that E. coli replicates every 20 minutes in the laboratory, but 12 hours in the colon. Because in the colon, E. coli has to face nutrient deprivation, it has to face competition from a large number of other bacterial species in the colon. And so it typically probably is in stationary phase most of the time. Bacteria that are in stationary phase actually are physiologically very different than the exponential phase cells. These are the cells that are much more resistant to antibiotics. These cells are much more resistant to being killed by phagocytic cells. So we, in the laboratory, test antibiotic sensitivity. We're usually testing cells in the logarithmic phase, but in reality, it's the stationary phase cells that are more resistant to antibiotic effects. So this stationary phase also can't last too long because since these cells are still metabolically active, some are dying, some are replicating, there is still an accumulation of waste products. There is still a decrease in the resources in the environment. So eventually they enter into the death phase here in 4, where you have almost like a radioactive decay. The number of viable cells are decreasing. There are still cells in here that are replicating. They are just exceeded by the cells that are dying. And actually, it's kind of interesting because you can pull the cells out at this point, and there's a genetic selection for cells that can do, in essence, cannibalism. So some of these cells are actually living now because other cells are dying. And bacteria don't have a conscience, so they don't have much of a problem consuming the guts of their dead brethren that have given up the ship and died. So there are still living cells in here for a while. So the one thing that I do like about the bacterial growth curve is that I think it's kind of a metaphor for humans here on Earth. You can think of us and our population in this way. We started off from very few people. We've gone through this log phase. Maybe we're still in this log phase, but eventually we're going to reach a point where we're living in enough of our own filth. We've used up enough of our own resources so that we'll hit stationary phase and eventually probably death phase. But that's hopefully many generations away. So just to kind of, so I did a quick graph of your dioxic growth curves from yesterday. And so Tuesday's lab sections collaboratively did this one and Thursday's did this one. And so these turned out really, really nicely. And so what you can see in both cases is there is a lag period here where the cells are replicating but not very much. And this is not on a logarithmic scale because I'm graphing the optical density at 600 nanometers on the y-axis as the independent variable. But then you see the log phase. And remember that, so we're going to see these graphs again later when we talk about the lac operon, because in reality what we're trying to do here is to show dioxic growth, and you guys showed it beautifully. Because what's happened here is that the bacteria, after they've acclimatized to this new media, this minimal media, where glucose and lactose are its only choices, it begins to grow very rapidly. As it turns out, and I've told you this already, that E. coli, given the choice of glucose and any other carbon source, will always use glucose to the absolute exclusion of the other carbon source until glucose is completely used up. Because glucose is the best, the most efficient carbon and energy source for bacteria. It fits directly into the glycolytic pathway. Lactose, on the other hand, the other sugar that it can use, has to now be energetically imported. It has to be broken down. It's a disaccharide, right? Glucose and galactose. It has to be cleaved into glucose and galactose. The galactose has to be converted into glucose to go into the glycolytic pathway. So there are other enzymatic steps that are required to use lactose. It loves lactose. It just can't use it as efficiently. And your experiments showed this beautifully. So what's happened here, actually maybe it shows up better in Thursdays. Here's the logarithmic phase where E. coli is growing like crazy. And notice that it slows down around 300 minutes. You get a second lag phase. So what's happened here is E. coli has completely scrubbed its environment of glucose, and it doesn't quite know what to do. It has to retool. We'll talk about this in the context of the induction of the lac operon and group translocation. But what's happening right here is that E. coli is now beginning to realize it has no more glucose. It's importing lactose, making all of the enzymes of the lac operon, and now hydrolyzing lactose. And then it takes off growing again. But the cool thing about this, and it shows up very beautifully, is look at the difference in the slope of the two logarithmic phases. Here you have a much steeper slope. The bacteria are replicating faster. Their population is doubling faster because glucose is such an efficient energy source. They're still able to replicate on lactose, but notice the slope of the line is less. They're actually having to invest more energy to break down lactose, so they don't have as much energy to replicate as fast. And so you can see this quantitatively in both of these graphs. These came out beautifully. We're going to come back to these graphs. You'll actually, here's a spoiler alert, you'll see these graphs on a lecture exam, but not for this first lecture exam, where we'll talk about things like inducer exclusion. We'll come back to the group translocation, but we'll talk about these graphs a lot more when we talk about the lac operon specifically. But great job this week. You guys did wonderful data collection. I know it was kind of a boring lab this week, but at least you didn't have to stay in the lab for 3 hours. So what I really want to talk about, so we could spend a lot of time talking about bacterial growth, how to quantify it, things like DNA replication. We're not going to do that. What I really want to talk about, though, are some of the consequences of bacterial growth. Because bacterial populations, as they change in numbers, have to change physiologically because their environmental conditions are changing as a result of their own numbers changing. So what we're going to do is we're going to talk about what arguably is the first evidence of multicellularity on Earth. So in evolution, We all think of bacteria as individual cells that are pretty much ignorant of one another. But in reality, many, perhaps most bacteria have the ability to sense and census their own population. So we're going to talk about a multicellular behavior that bacterial populations can do that affects their behavior. And notice the quotations around behavior. Now I'm referring to gene expression as behavior. We're going to talk about a process that I'm sure you've all heard about a little bit, at least in freshman biology classes, or I hope you have, quorum sensing. So if you think about the ability of bacteria to change, oh, I'm sorry, your question.
Is this within one bacteria's lifetime, or is this the result of evolution of the bacteria?
So is this within one bacterium's lifetime? So this evolved probably many billions of years ago, but this is a process that occurs in any bacteria's life as the population that it's contained within changes. So what's going to happen is that envision one bacterium not in a population, in isolation by itself. It divides into 2, it divides into 4, it divides into 8. That population now experiences different conditions, and so the bacterium, even that initial bacterium, will now change its physiology because the population has changed. And it makes sense if you think about it, because bacteria, and we talked about protein secretion at pretty good length so far, and you remember that bacteria are just wonderful secreting organisms. They secrete extracellular enzymes into the environment because they can't do phagocytosis. They can't bring large molecules in. So instead, what they do is they secrete enzymes like amylases to break down starch in the environment, proteases to break down proteins, lecithinases will break down fats, and then siderophores we talked about as a means to bring in iron. All of these molecules are secreted at great cost to the bacteria. But if the bacterial population is very low, and these bacteria are very small, one bacterium or 5 bacteria can't produce enough amylase, for instance, to have a significant degradation effect on extracellular starch. So they're not going to get a lot of return for the investment of producing that amylase until they've divided into a large population. If that population kind of uses its resources rather than to secrete these amylases, proteases, lecithinases, if it kind of does that only minorly but uses its energy instead to divide into 2, into 4, into 8, and eventually they get to 1 million bacteria, if all of a sudden that population looks around and goes, holy cow, there's 1 million of us now. If now we all begin to produce amylases at the same time, we can actually have an effect. And so this is what quorum sensing is. It's the ability of bacteria to detect that a quorum has been reached. The term quorum sensing was actually invented by a lawyer over Thanksgiving dinner. So one of the earliest people who discovered quorum sensing was a bacteriologist in England, whose name escapes me right now. But no, it wasn't in England. It was in this country because it was a Thanksgiving dinner. They don't do Thanksgiving. But I thought he, huh? No, she is a big name in that, but she did not discover quorum sensing. I think anyway, it doesn't matter who it was. He was trying to explain to his brother-in-law what kind of research he was doing, and so he was talking about this gene expression pattern that he was seeing when the bacteria got to large populations. And his brother-in-law, the lawyer, said, oh, it's as if the bacteria are sensing when they've reached a quorum. And he thought that was such a clever thing that he coined the term quorum sensing. And so basically that's what it is, though, is the bacteria reach a threshold population and they change their gene expression pattern as a whole simultaneously. So that's why I say this is an example evolutionarily of multicellularity. This is an example of a population of cells that you think of as being individual cells actually behaving as if they are a single organism or a single organ within an organism. So these are processes that would be very inefficient at low density. It's the same thing for pathogenic organisms. So this is another reason why it's important to understand quorum sensing. Because when any of us become infected with bacteria pathogens, we're generally infected with relatively low numbers of these microbes. Think of Streptococcus group A strep, which causes strep throat. If you become, and you've all become at one point or another, infected with group A strep and gotten strep throat, you probably got small numbers initially. If those small numbers of bacteria came into the back of your throat with guns blazing, producing every toxin that they can, being just maximally obnoxious, your immune system would kill them immediately. Our immune systems are fantastic at handling small numbers of bacteria. We don't do so good when the numbers are big. So what pathogenic organisms do when they initially colonize a naive host is they kind of try to fly under the radar. They don't express a lot of their virulence genes, a lot of their toxins, because they realize that if they come to the attention of the host can kill them because there's only 100 of them or 1,000 of them. But if instead they kind of lay low, hide behind the rocks, wait until their numbers increase to say 1 million or 10 million, then they realize that, okay, if we start to produce toxin now, we can cause all of the symptoms that are going to allow us to spread to another host. The sneezing, the coughing, the hacking, all of the things that go along with, exactly, thank you, right on cue. All of the things that go along with infectious diseases. And if the host begins to kill some bacteria, well, that's okay, because there's 20 million of them, as opposed to 20. So this is something that is very important in pathogens as well as environmental bacteria. So reaching this critical population or quorum. So one of the really fascinating things about quorum sensing, and it also points out how important it is to bacteria, is that it has evolved at least 2 independent times. Once in gram-negative bacteria and once in gram-positive bacteria. And there are probably some other examples of this. But so what we want to do, or what I want to do right now with you, is to go through a generalized scheme of quorum sensing in gram-negative bacteria, and then go through a generalized scheme of quorum sensing by a completely different mechanism, excuse me, that has evolved in gram-positives. And then we'll talk about a couple of specific examples with bacteria. So in gram-negative bacteria, where quorum sensing was first discovered, it's largely done, not exclusively, but largely done using a signaling molecule called an acyl homoserine lactone. These acyl homoserine lactones have a general body plan that looks like this, where R is an acyl or fatty acid chain. And that's important because remember that fatty acids or acyl chains like this are hydrophobic. So this molecule is going to have a hydrophobic end due to this acyl chain and a hydrophilic end due to this homoserine lactone. Now, there are hundreds of different species of gram-negative bacteria that are known to produce acyl homoserine lactones for quorum sensing. They tend almost always to be species specific. So species X produces a certain specific modification of this homoserine lactone that only members of its species can recognize. And it becomes largely ignorant to the acyl homoserine lactones produced by other bacteria. So to some degree, and there are obvious exceptions to this rule, but to some degree these tend to be very species-specific lock and key molecules. So these acyl homoserine lactones are produced by, unsurprisingly, an enzyme in the cytoplasm of the bacterium called acyl homoserine synthase. Acyl homoserine lactone synthase. HSL stands for homoserine lactone. This molecule, this protein, is produced at low levels all the time by every bacterium in the population when it's at low levels. And so when the population level is low, this homoserine lactone is producing homoserine lactones all the time, which now, without any energy requirement at all, can diffuse across the bacterial envelope. So because of that acyl side chain, that hydrophobic side chain, that's why that acyl chain is so important. It can pass across the cytoplasmic membrane, it can pass through the periplasm, it can diffuse out through the outer membrane and into the environment without any energy input from the cell. And also critical, it can come back into the cell the same way with no energy. So basically what happens is in this population, or in any population of bacteria producing these homoserine lactones, the concentration of the homoserine lactones outside of the cell is always equal to the concentration inside the cell. There's an equal movement out, equal movement in. There's no transporter involved, there's no energy production. So what this does is this is a perfect situation to allow a bacterium to sense the census, or to count the number of cells in the population. Because as the population of bacteria increase, so then does the concentration of homoserine lactones. And so higher concentration of homoserine lactones diffusing in now has the ability to trigger a cellular response, a transcription response inside that cell to change the behavior, to tell that bacterium that, okay, we've reached quorum, it's time to change our gene expression pattern. So the general term for the protein that mediates this is called LuxR, L-U-X-R, shown here as just a protein R. This is a response regulator. This is very similar to like the key B and key Y proteins that we talked about earlier. This is a protein that has the ability, in this case, to bind to DNA, but it can only bind to specific regions of DNA, and it can only do that when it's bound by a homoserine lactone. So as the concentration of homoserine lactone in the environment increases, indicating a higher number of bacteria, this pool of LuxR shifts from the non-homoserine lactone bound form to the bound form, shown as this shift from a circular molecule to a square molecule, the red molecule being the homoserine lactone. When it's in this particular conformation, it now has the ability to bind to specific DNA regions, specific sequences, to modulate gene expression. In some cases, it will turn genes off, but in many cases, it turns genes on. So in this very simplified model here, you'll see that the activated LuxR binds to DNA and now causes an increase in the transcription of this messenger RNA. This messenger RNA, or these species of messenger RNA, will include certain quorum-dependent proteins. These are usually, but not always, but usually secreted proteins. So these are the proteases, the amylases, the toxins, if this were a pathogenic organism. Because now the bacterium realizes that it's in a quorum, and so it upregulates the production of this messenger RNA for these quorum-dependent proteins, and the bacterium begins to secrete, or the population begins to secrete bacteria even more. Yes, Kirsten?
So is LuxR something that our cells produce as well, or is it specific to pathogens?
Good question. So this is the LuxR protein found within us? That's a really good question, because the answer is no. We don't produce anything like this. Because part of the reason why Bonnie Bassler is so interested in quorum sensing, and she actually has her own company that studies quorum sensing, is because if you want to design an antibiotic, a drug, to interfere with bacterial infections, you want to look for an Achilles heel, a point in which the bacterium has something that we don't have, and this is one of them. We don't do quorum sensing. So Bonnie Bassler and many other people are trying to come up with drugs to interfere with the quorum sensing system. Exactly. So that's great. So this system is uniquely bacterial. I don't even think archaea have anything quite akin to this. So you get this upregulation of quorum-dependent proteins, but then, and this is another important point because this is what really kicks quorum sensing into high gear. One of the proteins that's going to be increased by this quorum is going to be the acyl homoserine lactone synthase itself. So once the bacteria just reach quorum, they just get enough protein, homoserine lactone, to shift the pool more towards the bound form of LuxR. One of the first things that those cells do is they begin to produce even more homoserine lactone. So it allows the population, as it's reaching quorum sensing, to really ramp up very rapidly the homoserine lactone production so that the entire population gets the message nearly simultaneously. So these populations really act as if they've got a nervous system at this point because all of the cells in the population get the message almost simultaneously and behave identically. So it's very important that this homoserine lactone synthase also be what's referred to as a quorum-dependent protein, or a protein that's only expressed at high levels under quorum conditions. Yes, and that's just my last bullet point here. The homoserine lactone synthase gene also is controlled by this. This is, oh, did you have a question?
Yeah, so are AHL and HSL the same thing?
Yes, I'm sorry. Yeah, acyl homoserine lactone, and I'm referring, yes, I'm sorry, I used a different abbreviation here. Acyl homoserine lactone, AHL, same as homoserine lactone. Thank you for picking that up. I didn't notice that. I'll change that for next year. So this is kind of showing the same thing, but in a little bit more detail and a little bit more specifically. So here you have this homoserine lactone that can freely come in and out of the cell. It's being synthesized by this. This is the acyl homoserine lactone synthase. It's known by the general term based on the first gene that was determined to do this called LuxI. So LuxI, and I'm not interested in the biosynthetic pathway of homoserine lactone. This is just showing you that it's kind of a complex pathway where this enzyme goes through multiple steps, but eventually produces homoserine lactone. The homoserine lactone can come back in, and this, I like this slide because it actually shows, I think, more accurately the way the LuxR type proteins are activated. So it's not a matter of them going from spherical to square, as in the previous slide. In reality, and this happens a lot of times in the activation of transcription factors, is they become multimers. So these LuxR proteins exist when they're not bound to homoserine lactone as individual proteins that cannot bind to DNA. However, the conformation changes that occurs when homoserine lactone binds to them causes them to form multimers, dimers, or perhaps even more. And in this particular conformation, they can now bind to the promoter regions for target genes. And so these target genes would be the quorum sensing or quorum dependent genes, including the homoserine lactone synthase genes. Now, let's move on and talk about a general scheme for gram-positives. So as good as that system is, gram-positive bacteria have also evolved quorum sensing, but they've done it completely independently. And part of the reason why I like to emphasize this point is that it's a great indication to me of how critical quorum sensing is to bacterial populations. That bacterial populations have used completely different pathways to accomplish the exact same thing. They're not evolutionarily related. So in gram-positive cells like staphylococci, and we're going to talk about the specific example of how staphylococci do this, they have a different way of determining quorum. So here in a gram-positive cell like Staphylococcus or Streptococcus, there is a protein, a peptide signal produced in the cytoplasm. So this is not a homoserine lactone. This is a just legitimate full-on protein that's produced. And that protein is simultaneously chopped up into little peptides as it is exported from the cell. So this requires energy. This is a protein secretion pathway that is taking that signal peptide, breaking it up, and releasing all of these peptides into the environment. These peptides are analogous to the homoserine lactone, but chemically they're completely different. Homoserine lactones can go across the envelope easily with no energy. These peptides, once they're outside of the cell, are never coming back in. They are not going to come in to indicate quorum. But it is the accumulation of those signal peptides, just like the accumulation of the homoserine lactones, which is the indication that a quorum has been reached. The way the cells are detecting this quorum is rather than having these peptides come back in, they interact with a histidine kinase. You've already seen a histidine kinase, so you know something about how this works. We talked about key A. Remember that key A, when it receives the correct signal, let's say, for instance, a chemorepellent via the MCP protein, it can become kinase active on the cytoplasmic side. And that's what happens here, is that under low concentrations of these signal peptides, this specific membrane kinase, which has the ability to bind to this peptide, is silent. Its kinase activity is largely turned off. So what this does is it sets up a situation where over time, as the population of these bacteria, these gram-positive bacteria increases, and therefore so does the concentration of these signal peptides, now this kinase is much more frequently engaged or bound by its cognate ligand, in this case the signaling peptide. So the more frequently this kinase interacts with these, the more frequently its kinase activity on the cytoplasmic side is turned on. So this is another example of signal transduction, the signal being the peptides outside. That information is transduced across the membrane. Kinase activity is turned on. And just like key A, that phosphate group is transferred to a response regulator. But now, this response regulator, unlike in the chemotaxis system, is going to change gene expression pattern. The response regulators that you're studying for chemotaxis for the exam next week, those response regulators, when they're phosphorylated, change their ability to interact with other proteins. So this is partly a review for your exam next week. Remember that key Y, phosphate, interacts with the basal body to change the rotation direction. There's no change in gene expression. When key B, the other response regulator, becomes phosphorylated, it now has the ability to be an active enzyme to take methyl groups off. But there's no change in gene expression. The key A, key Y, key B system is very unusual among signal transduction pathways in bacteria. This is a much more frequent signal transduction system in bacteria, where you have the kinase being turned on, phosphorylating a response regulator, which now, in the phosphorylated form, is an active transcription factor. It can actually change the expression of messenger RNA. So in this particular case, in this general scheme, it's showing that it's binding as an activator in the promoter region to upregulate the expression of that quorum-dependent gene. So this type of signal transduction system modifies gene expression. These phosphorylated response regulators can also turn off other genes. And so there would be a whole series of spots on the chromosome where this phosphorylated response regulator would bind. So this is just another way, a completely independent way, that gram-positive cells have evolved the ability to detect quorum. So it shows how important that is. So let's talk about a couple of specific examples that are, I think, important for you to understand. So we'll stay with the gram-positive. Maybe I should have done the gram-negative first, but since we just talked about gram-positive, let's talk about Staphylococcus. Because Staph aureus is a really important pathogen. And its pathogenicity is so incredibly intertwined with its ability to do quorum sensing that really they become avirulent if they lose the ability to do quorum sensing. So the way Staph aureus does this is it has this complicated system where it produces this protein, and again, I'm not interested, I will never ask you a question on an exam like, what is AGRD? I'm not interested in that. I'm interested in the overall thing. So if I mention these gene names, don't feel that you have to know them for exam purposes. But this protein, AGRD, produced by a gene here in this operon, will be proteolytically cleaved as it's exported by AGRB into these autoinducer peptides. AIP stands for autoinducer peptide. And unfortunately, they make them look like homoserine lactones or sperm cells, depending on your point of view. I tend to look at that as shorthand for homoserine lactone. But these are peptides. So they are not going to come back into the cell. These autoinducer peptides, as they increase in concentration, more frequently engage this histidine kinase called AGRC. So this AGRC has domains that are exposed to the extracellular environment that have the ability to transiently bind to these autoinducer peptides. The more frequently that occurs, the higher the rate of kinase activity on the cytoplasmic side. So here's that histidine molecule, a histidine amino acid that becomes phosphorylated. That phosphoryl group from AGRC is transferred to AGRA, also a member of this operon of genes shown here. AGRA, in the phosphorylated form, now becomes an active transcription factor. So now, and only now, will you begin to get the expression from this piece here stands for promoter. So promoter number 3 is a promoter that's driving transcription to the right through this gene called RNA3. Now, it's outside of the scope of this particular lecture. We'll talk about regulatory RNAs, I hope, later in the class. But for the moment, just realize that now the expression of this RNA3, which does not encode for a protein, so it's a regulatory RNA. This regulatory RNA now has the ability to simultaneously, at different sites in the chromosome, increase the production of exotoxins by the staph and decrease the production of surface proteins. So basically, this bacterium, once it reaches quorum, kind of becomes like a stealth bacterium. It begins to stop the expression of proteins on its surface, So there's less targets for the host cell to recognize it as foreign. And at the same time, it begins to pump out a lot of toxins to kill cells like white blood cells that might want to kill the staph. So this is a really highly evolved ability of this bacterium to be, this is part of the reason why it is such a serious kick-ass pathogen. Now, people are actually trying to take advantage of this in the pharmaceutical industry. So this is why quorum sensing is so important, because I'm hoping, and I think a lot of people are hoping, that it's going to be, quorum sensing is going to be another Achilles heel of bacterial population. So what some people have done is they've come up with an antibody. So this little blue triangle here, and I don't know if you guys have had immunology yet, we'll talk about antibodies a little bit later. But this is a molecule that can be synthesized from B lymphocytes, and we can make monoclonal antibodies against them. But in any case, pharmaceutical companies have come up with an antibody against this autoinducer peptide. In this slide, it's shown as this little red cannon-looking thing. So that what's happening in staph is that at low population densities, when quorum sensing is off, the bacteria are expressing adhesin genes, which allows them to colonize the host. So these adhesin genes bind things like fibronectin and collagen, things in our connective tissue where staph loves to live. Staph will actually infect virtually any organ in the human body, from bones to brains to skin to kidneys to livers. It is a really bad pathogen. But when it's first colonizing, it's expressing lots of surface proteins because it wants to adhere. And it does that because quorum is not reached, so there's not much autoinducer peptide. Once the colonization occurs and the population begins to increase, they reach quorum. So now quorum sensing is turned on. They begin to express those toxins and other virulence determinants. And they also begin to stop producing those adhesins. This allows them to become invasive. They can actually go through the connective tissue, get access to the bloodstream, spread throughout the body. It's a really evil, highly evolved system. You couldn't draw a script better if you were a Hollywood screenwriter. So what people are trying to do is to basically trick staph to think that it's not at quorum. If you can either use these, I don't honestly understand these apolipoproteins having the ability to bind, but antibodies make more sense to me. I understand them better. If you put in an antibody that can bind up and sequester those autoinducer peptides, you can convince that large population of staph that it's not at quorum yet. So it won't start producing the toxins. It won't stop producing those adhesins, so it won't become invasive. So this is another way that we can potentially intervene in the infectious process. Okay, and so now let's talk about an example in the gram-negatives. And this is really where quorum sensing was discovered, in this particular bacterium, this charismatic Vibrio fischeri. This is a marine bacterium. It's related to the bacteria that causes cholera in us, but this is a completely non-pathogenic bacterium. This is the bacterium that actually Will Soto, new faculty member in the biology department, works with. So it's a host-associated gram-negative bacteria, but you can isolate it from free ocean water. And it's associated with certain fish, but especially associated with squid. A little tiny squid called the Hawaiian bobtail squid, which is absolutely adorable. But it exists in a mutualistic interaction with the squid. The mutualism is based on the fact that this bacterium is bioluminescent. It has the ability to produce light, but it only produces light when it's at quorum. Because if you think about it, it's like the old Zen cone. What's the sound of one hand clapping? If you're one little tiny bacterium and you're producing light, you're never going to be able to produce enough light for anybody to see it, and it's very expensive. So don't produce light, don't produce luciferase when cell numbers are low. Wait till there's a lot of bacteria. Then if you all simultaneously begin to glow, that's a visible light. And so that's what these bacteria have. The Vibrio fischeri has this operon of genes called the lux operon. This is where the terms lux-I and lux-R come from, because the initial studies, the initial discoveries, were done by a guy at the University of Southern California who was studying the system. And for the longest time, people thought, this is a great system, but it's only found in this marine bacterium. It can't be wide, and it's very widespread. But in this bacterium, at low cell density, they're not producing a lot of this homoserine lactone, so this is just the chemical specificity of this 3-oxo 6-carbon homoserine lactone. It doesn't produce very much of it. But then at high cell density, with lots of that homoserine lactone coming back into the cell, it binds to the LuxR molecule, now causes it to dimerize and bind and produce a lot more messenger RNA and hence proteins. And so the bacteria will now, only at high concentrations, produce light. Now, what's the advantage to light? To the bacterium, there is no advantage to producing light. It's tremendously energetically costly. So the bacteria only do it to pay the rent. So what these bacteria do is they live in a mutualistic association with these adorable little squid, the Hawaiian bobtail squid. So let's start at night. So it's a nocturnal predator. And calamari is delicious. So there's lots of predators in these shallow waters in Hawaii that would love nothing better than to take a bite of this calamari. So what happens as they're hunting at night, they have a light organ on their ventral side that is filled with these luminescent bacteria. This is the process known as, or a predator avoidance strategy known as counterillumination. They basically are producing a moonlight or starlight, so that when that squid swimming through the water passes between a predator lying below and the moon or the stars above, it doesn't cast a shadow. These things are so well developed that that light organ is within the ink sac. So you all know that squid, remember squid can eject ink, like octopus can. That ink sac will change with the phases of the moon. So during a full moon, the bacteria will produce more light because that ink sac is more open. But when the moon is more towards a quarter moon or a sliver of a moon, it closes it up more so it's not producing quite as much light. So it's really highly evolved with this squid. So this squid hopefully makes it through the night without getting eaten. And then it buries itself in the sand to hide during the day. Now, that light organ has muscles around it. The squid will literally contract those muscles and expel about 95% of the bacteria because now they're like your E. coli culture looked this morning. Really pretty sick. Used up a lot of resources, produced a lot of toxins. So the squid just expels as many of those bacteria as it can. And then over the course of the next several hours, it allows those bacteria to grow slowly. They increase in population, they increase in population. And guess when they reach quorum and begin to produce light? Exactly at sunset. So the next night, when they come back up, the bacteria have just reached quorum just before sunset. So now the squid can hunt safely again and just repeat this over and over again. Really beautifully, highly evolved system. And I have a video on it, but there's only 3 minutes left in the lecture, so I'll take a question and then we'll look at the video when we come back. Yes?
Sorry, I was just wondering, like, 95% of the bacteria die. What is their benefit of being in a mutualistic relationship if they have that high of a chance of being expelled and killed?
Good question. So I should have made it clear that when those bacteria are expelled, they're not necessarily dead. So they're going out into the environment where they can survive and colonize new little baby squid later. That's a good question. Okay, otherwise I'll see you guys on Monday. I'll see you next month, right? October. Have a great weekend.